If you can, would be great. Guys, hello. They leave once I speak. You coming to the lecture?
Yeah.
Yeah, come on, quick, quick, quick. He's going to start.
Professor Darzi?
Yes. Okay. Is this working? Yeah, great, thank you. Well, distinguished guests, ladies and gentlemen, good afternoon and welcome to Imperial College London. Some of you may know me, most may not. Darzi is my name. It's my great pleasure and privilege to introduce one of the greatest innovators alive today, Dr. Noubar Afeyan, CEO of Flagship Pioneering and co-founder and chairman of Moderna. And I suspect some of you would have had that jab in the last year or so. Noubar has dedicated his professional life to improving the human condition through scientific innovation, working at and beyond the boundaries of what's possible. We tend to train some of our students in that mind thinking, but you have the real person here. He has numerous scientific publications and is the inventor of more than 100 patents. During his career as an inventor, entrepreneur, and CEO, Noubar has co-founded and helped to build more than 50 life sciences and technology startups. Just around the millennium, he founded Flagship Pioneering, a unique organization that conceives, creates, and scales these companies. At Flagship, entrepreneurial scientists are encouraged to think beyond the boundaries of what is possible today, and to ask the question to themselves, what if? And then they seek to answer that question. It is precisely because Noubar has the courage to think beyond today's adjacencies that Flagship has achieved this remarkable breakthroughs. And it starts with the belief that it is possible to change the world. Whether hypothesizing that red blood cells could carry a payload as well as oxygen and providing that they can, or believing that mRNA, the factory forearm of the cell, could be harnessed and making it happen. He has recently launched a new initiative at Flagship called Preemptive Medicine and Health Security, and he's asked me to chair that, and that imagines a world liberated from disease. If anyone else said that they believed they could forge a future where conditions like diabetes were consigned to history, then they would be dismissed as a dreamer or even a fantasist. When Noubar says it becomes a real possibility. The results of thinking big has been a creation of a series of first-in-category companies with extraordinary impact. By conceiving and creating scientific platforms, the breadth of application exceeds anything found in traditional pharmaceutical research. The most famous bioplatform company known to us, obviously, is Moderna. At the beginning of the company's journey in 2010, it is fair to say that many greeted the idea with skepticism. Indeed, Moderna's own CEO, when he was appointed, Stefan Bancel, said that his first reaction was, "You're crazy. It's never going to work." Noubar, of course, proved them otherwise. After 10 years of experimentation and refinement, and more than $5 billion of private investment, $5 billion of private investments, Moderna was able to design its COVID-19 vaccine just 2 days after receiving the genetic sequence. And just 2 months after that, the Moderna vaccine was the first to go into human trials. And within a year, 1 billion doses have been manufactured, saving lives around the world. It is a remarkable story of innovation, and Noubar's personal story is as remarkable. His family, like mine, are Armenian. In the wake of the Armenian genocide, his parents settled in Lebanon, where Noubar was born. When the civil war broke out, the family moved again, this time to Canada. He memorably describes innovation as an intellectual innovation. In other words, taking your mind to a new place and imagining what could be rather than focusing on what once was. It is not surprising then that Noubar was awarded a Great Immigrant Honor from the Carnegie Corporation in 2016. Noubar came to the US in the 1980s and entered biotechnology during its emergence as an academic field and industry, completing his doctoral work in biochemical engineering at MIT in 1987. He was a senior lecturer at MIT Sloan School of Management from the 2000 to 2016, a lecturer at Harvard Business School until 2020, and elected to the National Academy of Engineering this year. He teaches and speaks around the world on topics ranging from entrepreneurship, innovation, and economic development to biological engineering, new medicines, and renewable energy. Together with his partners, Noubar has launched philanthropic projects including the Aurora Humanitarian Initiative, FAST Foundation, and the WC Diligence School. He is a member of MIT's governing body and sits on the board of trustees for the Boston Symphony Orchestra. And if you're wondering how he finds the time, it's because he works more or less 6.5, 7 days a week. So ladies and gentlemen, without further ado, please join me in warmly welcoming Dr. Noubar Afeyan.
Well, thank you so much. With an introduction like that, I'm tempted to simply take questions and answers because I can only disappoint you from there. It's a great pleasure to be back at Imperial College, where I had the distinction of addressing an audience some 7 years ago, it seems now. And at the time, I did a little bit of what I want to do today, which is really give you a cross-sectional view of the various frontiers in which our teams are operating today, hoping to paint a picture of the future. Back then, I did describe mRNA as a new platform, and I'm sure it was highly incredible at the time. And so today, I'll try to describe some other things that you might find warranting of equal skepticism. And I hope in our Q&A period, we can discuss those and discuss what about them makes us excited. So I've titled this talk Emergent Discovery because one of the things I'm going to start out with is trying to tell you how we think about innovation a little bit differently than what might be found in the traditional sense of the word. And much of what I saw already today in my tour suggests to me that a lot of what's going on here equally is thinking about making pretty significant leaps and then filling the gap. And so I'm very encouraged that there will be some level of resonance, hopefully, in what I'm describing and what you're doing here. So let me start out on that theme at a very high level. You know, the world, whenever it gets in trouble, whether it's climate change or food insecurity or any number of things, turns to innovation as the answer, and better yet, breakthrough innovation. And breakthrough innovation is viewed to kind of be this special category of innovation where major disruptive change comes. It might have been search engines a couple of decades ago. It might have been digital photography 40 years ago. It might have been biotech drugs 30, 40 years ago, et cetera, et cetera. And the way the world thinks about these breakthrough innovations is a matter of placing a lot of bets without being able to know in advance which ones are going to actually work, and then judging them early and often, and discarding the ones that don't look promising and advancing the ones that do. And of course, we all know that much of the greatest innovations in the world weren't born in perfect shape. They actually were pretty ugly, deficient versions of themselves in their original embodiment. And so much of what could become breakthrough innovations, we discard very early on by the process by which we go about doing them. In fact, we ask experts, we ask leading thinkers to judge these at their earliest form. And of course, they are going to focus on all the reasons why not to believe them. And so this notion of breakthrough innovation as a shots on goal strategy, the good example of that is the pharmaceutical industry. Pharmaceutical industry over time has become largely a shots on goal, that expression that comes from hockey or soccer. If you don't take shots, you can't score, therefore just take lots of shots on goal. And that kind of, for a practitioner, where you're really just working on one shot at a time, is a very disappointing attitude that you're kind of part of this screening experiment, if you will. So the question is there any other way of thinking about this? And of course, we are coexisting with a non-man-made version of innovation, which is called Darwinian Evolution. And it's been around a lot longer than we've been around, and it's produced some pretty marvelous innovations. Various species, various organ systems in our bodies that far exceed any man-made innovation by any measure, in my view, whether it's in its microscopic kind of precision, whether it's in its complexity of interaction, you name it. We have not, as humans, created things that are nearly, in my view, as complex as what nature has. But nature didn't do that by shots on goal alone. It basically has this notion of descendancy, which is that partial solutions lead to better solutions. And this transmission of that information, otherwise through our genomes and through other epigenetic changes, is an incredibly important part of the learning process that leads to better and better versions of descendant things that ultimately will gain advantage. And that thought is relatively absent in the way we think about designing for breakthrough innovations. But in fact, if you study the history of innovation, you find ample example of not only evolutionary kind of processes, but the way they're even described, where things are related to other things, and people say, I took this idea or this gene from one place and I applied it to this. So we're doing this, but we don't think about it in a formal way. So I guess part of what I would urge you to consider, and part of what Flagship is entirely based on over the last 20 years, is this notion that you can think about Darwinian evolution as a mechanism by which to actually come up with great derivatives of otherwise bad starting points that lead to breakthrough innovations. And I'm going to give you some examples of that. So we call this kind of emergent discovery. About a year ago, after practicing this for 20 years and not saying much about it, largely thinking that it's probably not going to work, and it might still not work in its full embodiment. In other words, I'm not here to kind of prove to you that this is in any way superior. It's just an alternative way to think about it. We published a small article that laid out the logic of this. So any of you who are interested in a non-scientific publication on strategy, on kind of innovation strategy in the Harvard Business Review, there's an article that you can refer to that describes much of what I'm going to say, at least at the conceptual level today. So what do I mean by Darwinian evolution? I mean simply variation, selection, and iteration. I don't mean DNA, I don't mean RNA, I just mean the concepts of variation, selection, and iteration. Now, you might be thinking, how much variation? Who does the variation? How much selection? Who does the selection? Do you weight it differently? And iteration. And the answer is, we had to kind of figure that out over time, right? There's no recipe we could read. We could not look at, various components of what nature does and try to port them literally. But the concept that it's really about conducting, if you will, like a conductor conducts an orchestra, you could think about breakthrough innovation as an emergent result of conducting an iterative set of variation and selection processes. And that's kind of what we lay out here. And that, by the way, has profound impact on culture, because we as humans reward breakthrough innovations as though the people who were there when they finally came to life are geniuses of some sort. But in fact, if you believe it's an emergent process, the people who were there when it came about are participants in a long descendancy process. They just happen to be able to describe it in whichever way they'd like. And I've met very few people who will describe it quite the way I am, which is that they're a conductor. And just like a conductor doesn't make a single musical note, they too often don't make a single musical note. But they result in the music that gets produced. And in my view, music that you hear in a symphony or the result of a breakthrough innovation are emergent, deeply emergent. That's at least my belief system that I'd like you to have a small piece of your mind dedicated to, even while you do it the conventional way otherwise, because I recognize that what I'm saying is quite unorthodox as it relates to funding. If you went to somebody and said, I think I'm going to have a lot of bad ideas that eventually are going to lead to good ideas, give me money, most people won't give you any money. Our grant-making process completely forbids this type of innovation. If you went to people and said, join my team, let's come up with really bad ideas that lead to, people won't leave their jobs to join you. And yet, if I'm a little bit right, I would argue to you that much of the breakthroughs come about from that. So we're pouring a lot of resource over here, but we expect these breakthrough innovations that often come from over there. I just think it's worth thinking through. So I'm going to make one other conceptual thing, and I'll give you some examples of how we're thinking about it and where it's being applied today. The other thing to think about innovation is where it's taking place. And I don't mean by that in which scientific discipline or technology, but just generally think about whatever exists, whatever is known, and fit it into this circle that's drawn in the middle called current solutions, current knowledge. And that's the zone of kind of incumbency, right? Experts dominate there, large companies who own a field dominate that zone. And if you're an innovator or a scientist, you're trying to break into that, but they occupy that, they preoccupy that long before you could get there. And where does innovation happen vis-a-vis this gray zone? And that is, I would call, in adjacencies. There was a notion some 20 years ago that Stu Kaufman, a theoretical biologist who wrote a lot about evolution, put forward about evolution, which was that it's a measured sampling of adjacent probables. So evolution also has this tendency to kind of sample what's within a short evolutionary distance from it. And so his notion was that. And I've come to view somewhat evolution as being done in this way. From any given point of time, you're sampling the adjacent possible. And by placing a lot of bets in that adjacent possible, you're essentially expanding knowledge, and sometimes utility comes from that. And that's kind of this zone of adjacency that I'm pointing to. Now you might say, what defines this distance of the zone? And I would argue the distance of that zone is the distance over which people are willing to make risk-reward evaluations. You know this, because if somebody comes to you and says, I'm going to make a drawn fiber, as I just heard about, that's going to go in the body, but then I'm going to take it out, put it in again, I'm going to do this. At some point, you'll say, look, that sounds too much like fantasy. I can't even tell you what the risk-reward of that is, because you lose your confidence of having it be relatable. So just consider that now outside the zone of adjacency. And against that backdrop, what we were interested in is saying, could we knowably invent far outside the zone of adjacency? In other words, work on things that are untethered, unconnected with what the current scientific dogma or its extrapolation is willing to accept as possible. That's just a choice you make, right? It doesn't, clearly, if you go back 100 years, the zone of adjacency would be right around the center of this circle, and everything in that is current knowledge would have seemed fantastic back then, and yet Fleming, others that you hear kind of are descendants of, clearly said, no, actually, you can do these things outside. And so we know that this outside zone is going to be occupied over time. We just can't guess today which ones to go after because we don't have the safeguards of adjacency. Does that make sense? So the question is, what do you do then? And that's kind of what we've been thinking a lot about and practicing over the years, because it stands to reason that a subset of these red dots way outside might be valuable. If you could only figure out which ones and accept the fact that you're not going to be at all confident about which ones because you don't have anything to go by. And so that you need different mental models to deal with it. So how do we operate in this space? I'm going to tell you in a nutshell, and then I'll tell you the results of some of these things. So we, over the years, have developed our own process for how to think about innovating and then creating companies around these things. And everything starts, I'm going to use words that are kind of probably a bit provocative, if not disorienting. I'm going to start on the left side of the slide by the words leap of faith, right? And I use that very carefully. From a non-religious standpoint, just look at the definition of faith. In general, is belief without data. That's faith. And it's often used in a derogatory way in the sense that you're not supposed to use faith in science. You're supposed to use proof and fact. But we choose to start with leaps of faith. We ask, as Ara mentioned in the beginning, what if questions. What if there were viruses in our body that are essential to our health? Not pathogenic, but essential, that we depend on for our health. Now, I don't know if anybody here studies such viruses, but when we asked this question 4 or 5 years ago, we were told something between there are no such viruses or nobody studies such viruses because they're not pathogenic, because the funding goes to pathogenic viruses. In fact, virus is not even a word anymore in human health. Pathogenic virus is the word. It's like one long word, because that's the way we conceive of them. And if you wait till accidentally somebody finds non-pathogenic viruses who they get money to study, even though it's not good for anything, you might eventually realize that there are actually tons of commensal viruses, And immediately those in the room who might have run into microbiome research, as I visited a while back and I saw quite a lot of work here, you'll say, of course there must be commensal viruses because there's commensal bacteria. What stood in the way of a virus becoming a symbiont with humans? And you could talk your way into imagining that, in fact, that should be possible. In fact, how could it not be possible? Well, that mental process is making a leap of faith. You first make a leap of faith, say, you know what? I think they exist. Now, where would they be if they exist? How would I find them? What would they be good for? Who studied them? And if you work backwards, that's literally the point of this, it gives you hypotheses you would not otherwise make if you're staring at the world from the current present, the literature that exists, and everything that is a responsible, safe distance from where you are today. That's what we mean by leaps of faith. So these generate seed hypotheses for science that is not really provable, products that are not at all existent, often needs, medical needs that you can't really yet point to, but you got to start someplace. And so we create these hypotheses, and these hypotheses then allow us to engage in the most exciting part of the journey in the beginning, which is we go to people who are knowledgeable in these fields, and we share with them our completely, initially, fantasy ideas. Now, these are not arbitrary fantasy. They are absolutely safeguarded by a scientific description that makes them seem plausible, right? And if you're sitting in the audience, and I'm saying this in a bit more of an exaggerated way, so forgive me, but you think science fiction, right? What's the difference between arbitrary fiction and somebody who's trying to do science fiction? And often they'll take the time to make the science seem credible, even though there may not be a worm man or a mitochondrion man, but nevertheless, you kind of give it some reasonableness so people can relate to it, right? And of course, many of us have seen that over the years, a lot of what is written in science fiction 50 years ago ends up becoming science or technology that many years later. Why? Because somebody took the time to envision things that were at least consistent with some laws of physics, etc. So we do some of that. It's not like we're saying we're going to fly to the moon and back in 3 seconds and cure this, and all of a sudden people go, well, you know, so you got to kind of think about the idea described in a way that at least is understandable, even if not provable. That's kind of the key to the beginning. But here's what we do. We go to people who are experts, not just narrow experts, but also some that are have broad expertise, and we share with them our ideas. We want to develop a viral vector to deliver genes into the body using a human virus. And then they'll say, which human virus? We say, we don't know yet, but just suspend disbelief for a second. We're going to do that. And they say, people tried that 20 years ago, and here's what they found. All of a sudden, what you get into is that people will start applying selection pressure, driven by their utter disbelief of what you're saying, but where their expertise is now raining upon your ill-formed ideas and informing them. To which you can either become completely intimidated and walk away and take your idea and never say it again, or you can be the change agent, which is you iterate your idea. And you iterate your idea conceptually until you overcome these otherwise spontaneous objections, only to again go to another person and share your idea. It takes a lot of self-confidence and humility to do this, by the way, because you expose yourself to experts in a field who are going to think you're crazy. But if you share with them that this is a process you're trying to undergo to try to find things worth working on, what we've observed over the years is that, in fact, people will engage, because that's a part of their thinking they're almost never allowed to use, right? Even in the most, kind of extreme grants they get to review or papers they get to review, people aren't allowed to go there. And so this is a little bit of kind of their entertainment, if you will. So just encourage them to be useful. A few generations of these discussions later, before we do a single experiment, we'll find a descendant form of the idea that actually overcomes most of the objections that people are coming up with, or you discontinue it. If you can't overcome the objections, you don't have to waste time going into a lab and trying to see if something could be made to work. So that process, which is described here in this first phase, what we call exploration, leads to ultimately refined hypotheses that after many, many discussions, you can't find out why it shouldn't be a good idea, except nobody's done it yet. Then and only then do we go into the lab in the second phase, the proto-company phase, and we try to reduce the idea to practice. And that's an interesting set of words, reduce to practice. If you've been involved in patents, you'll realize that a patent has to demonstrate reduction to practice. But in the way we think about science, we don't think of reduction to practice, because we're doing exploratory, knowledge-seeking work, as opposed to asserting something can be done and then try to figure out how to do it and then going backwards. So that reduction to practice is a different type of experiment you need to do. Still, it could be falsified. Hypotheses could be falsified. You might fail to prove it. But if you succeed in demonstrating the elements you need to believe something can be done, for example, in my virus example, that such viruses exist, that you could find evidence they get transmitted between humans. Turns out all of this is done. I'm not making up this example. It's a 3-4-year-old project we did where you've really, what would you do if you wanted to find a non-pathogenic human virus? You'd go look for a tissue. What would you look for? You don't have the sequence. So what you do is, it turns out, you'd look for DNA sequences from humans that have been sequenced who don't fit into the human genome, the canonical genome sequence, because these ones, they're discarded. They're junk DNA. Turns out they're pretty much viruses. Then you take those and you do pattern recognition to the kinds of ORFs and other kinds of parts of viral sequences, and you find that there's a whole bunch that match, except they've never been made. So what else do? You go look for those sequences being transmitted from one human to another. When? Upon blood transfusion. Because when you have blood transfusion, if there was a sequence in the donor and a sequence in the recipient magically appearing and persisting, that's probably a virus that the body's not getting rid of, and that is potentially commensal. That's exactly what our team did. And we identified that in a class of virus, which was known but not really researched, called anelloviruses, you could find a starting point that then led to the rest of what you see here, which is reduction to practice. We actually made the virus, we vectorized it, and then you create a platform. I'm not going to have time to describe all of these details, and that's what a company focuses on, is really focusing on creating the combination of pieces you need to be able to generate multiple products. And then finally, that results in a venture. So I've gone through a deeper description of this than I expected, so let me kind of use the rest of the time to give you some examples. Moderna is an example that started around 2010. And our habit is that when we do these explorations and we prototype them, we number our companies. So Moderna actually wasn't born as Moderna, it was born as LS18. It stood for Life Science 18. It was the 18th such project we did. And the reason is, it turns out that it's a lot easier to discontinue a number than your favorite name, Phoenix Rising. Or so you call your company something and it's a disaster to say, whoops, didn't work. So it was a number. And we started asking questions like these. This was not following the research that had been done previously, because the research that had been done previously took on a very different meaning once we decided to make therapeutics out of RNA. The reason that had been done previously was fraught with limitations and challenges and innate immune responses and all sorts of things that people in the RNA field had observed. Suddenly we said, no, we're going to make drugs. Now what do we need to believe that can be done? So we asked the question, what if humans, patients, could make their own drugs? That was the original thought that led us to say, well, how would you do that? If you wanted to insert a piece of code, what would it be? Would it be DNA? Would it be RNA? Would it be a virus? Would it be proteins? They all can provide some level of information. And what if you could do this in a transient way? Because we were quite concerned, if you go back to 2010, at the then apparent peril of gene therapy done through DNA through integration in random sites. And that concerned us then, concerns us somewhat now, but we've been seeing progress. But back then it was a big concern, because gene therapy using viral vectors was not something that people were being able to be reliant on in terms of making genetic manipulations. And so with those types of questions, we set out on a journey that now, 12 years later, and it's been well covered in the last couple of years through no fault of ours, basically because of COVID, but now has a company that has 46 different clinical medicines under development, and roughly 27 of them undergoing human clinical studies, of which 2 relate to COVID. The remainder relate to many other diseases. So, how does that happen? Well, this is a slide that's now, I think, been on Vogue and Sports Illustrated, so I'm not gonna tell you this central dogma of biology, but we used to back in the day, because people didn't really remember high school biology, so we had to kind of say, well, look, we're interested in going from this, the center molecule, to a way to get it inside cells, such that we could essentially, using the ribosome, by the way, not that unlike what viruses do, make, in our case, whatever protein we wanted. Not more of the virus, but whatever protein we wanted. And this is a drawing that we've used for probably 7 of those years. And it's kind of led the way to how we've thought about things, which is you can get things to a nucleus, you can get things directed to the mitochondrion, certainly cell surface proteins, secreted proteins. So we had a broad vision of how this could all play out. And so we followed that. And this is a slide, it's a bit of an eye chart, that kind of lays out the transitions we went through. Back in the day, in 2010, this was an internal project in our labs. We filed a set of provisional patents that described all the things that would lead us to a therapeutic platform, and for that matter, a vaccine platform. And then we began to reduce it to practice. And along the way, you'll see names like DARPA, a major defense granting agency, and AstraZeneca, and Merck, and BARDA, and many other folks who early on got involved in providing complementary resources. Could be capital, could be expertise, disease expertise, could be other things. And that was an important part of the journey. And what you see all the way till about 2015 is that we had no clinical activities. In fact, ironically, the first clinical trial we ran, 2 of them in parallel in Germany, was vaccines against potentially pandemic influenza strains, which people don't realize, because in 2020, when they first heard of Moderna, they thought just then we had started working on human pandemics, let alone human viruses. And in fact, it turned out that COVID was the 10th virus against which we had tested vaccines in humans, not the first. And the first was back in 2015. And what we wanted to do actually back then is to pick strains of bird flu that had not entered humans before, such that when we could just see an immune response without risking any other background effects, et cetera, we just thought, this is a very clean signal. And we were going to use it for stockpiling for future vaccines for preparedness. We did not expect that 2-3 years later, at least in the US, stockpiling would be out of vogue and nobody was interested in this. But the data initially showed very encouraging results. We then went on and worked in cancer. You might say, why go through that? Well, personalized cancer vaccines are one of the most interesting uses of mRNA technology. We started working on this more than 6-7 years ago with Merck. And what was interesting for that is that it turned out that's what positioned us well to be able to move so quickly in the pandemic. Because we had already started a human trial where we were taking patient-derived tumors, sequencing them, computationally deriving potential neoantigen sequences, creating a purely computationally designed mRNA sequence that had 30 and eventually up to 50 antigens that are presented for T-cell activation in a single construct for that patient, manufacturing it, putting it in LMPs, putting it in a vial, and sending it for a patient to receive a self-vaccine against their own cancer, all in 2 weeks. And that manufacturing line had already been established and was running, and we had already gone into 40, 50 people using this. Well, it turns out that when the virus showed up, our manufacturing people said, I know how to make one of these. I do it every day. Just don't call it a cancer, call it a new virus. And I can take the sequence. I don't have to even come up with 50 different antigens. Just come up with one, the spike, make that, put it into an LLP, and go. And so it's interesting how a platform allows you to manifest the technology in so many different ways. This helped us a lot. So that's why 2017 was a big date. 2018 was a big date for us because we had built a manufacturing facility which was anticipating a scaling up of this cancer program, not at all an infectious disease program, because we were many years earlier from that. Well, then COVID happened, and a lot of this underlying science and the type of investment that we had made building this platform was directed pretty much overnight to saying, could we enable at least a quick proof of concept that such a thing can be made, a vaccine could be made against an unknown virus at the time. And as was mentioned, within a couple of days, we had what is today the final product in its full sequence form, designed and made. We started testing it in humans. Then we did a very extensive test, which you see in the middle, as I think many of you are well aware of. And by November and then ultimately in December 2020, we obtained emergency use authorization. And eventually, you've seen additional work done in boosters. Now we're doing variant vaccines. And I'm happy in Q&A to answer anything you might wonder about this. But for the purpose of this talk, this is kind of the journey disembodied. Now, what I'm going to do from here on is to give you non-Moderna examples of the same journey, because they all kind of started out with similar what-if questions. And the what-if question of this project, which is now currently known as a company called Generate Biomedicine, it's about a 4 or 5-year-old company now, started out by us asking the question, what if we knew the DNA code that codes for any arbitrary protein function? So could you, at the DNA level, specify a beta-lactamase? Could you specify a anti-PD-1 antibody, purely at the DNA level? Or do you need to make the protein, look at its three-dimensional structure, or use AlphaFold to predict it, and then compare it to which comes first, the DNA sequence or the function? And of course, we know that the DNA sequence must embody the function because that's the only thing that gets transmitted. But we don't know how. And so the question was, could we, using now machine learning tools that are, let me just go back, I think this is going to run, you'll get this. We literally looked into, it's not, the slide's not progressing, but anyway, you're probably all aware that there are, in the generative AI field, people are generating human faces. We knew that 4 or 5 years ago. And people are doing all sorts of things that are essentially tying ultimate end results back to the underlying components. So we thought, could we do this using machine learning, which has shown enough examples of what it is you're trying to make, that it essentially learns the underlying sequence information to recapitulate. And so that's what we call generative biology. It's basically using a substantial amount of data to train models that can truly computationally generate a protein of arbitrary function, that's the dream, or a relatively straightforward function, which is, I want to bind to this protein here. That's a function that has huge therapeutic value. That's what most biotherapeutics, antibodies, are about. So the question is, could we do that? And when we started out, you couldn't. There was no proximal approach that you'd say, yeah, if you just put more money into this, you could now imagine that. You had to go into labs, do large evolutionary experiments using phage or yeast display, et cetera, but you could not sit there in front of a computer and do this. And so we did not know if this could in fact be coded or decoded, if you will, back into the DNA sequence. But we thought, well, guess what? Our ability to synthesize things is relatively advanced and cheap. Our ability to take data and train models was getting dramatically better. And so could we try this? And in fact, that's what Generate has done. I'm just showing you slides that lay out this kind of approach. And so what have we done? Several years of trying later, we now have, we believe, the ability to make literally computer-generated antibody sequences at the DNA level with very specific binding sites with no more information than which site you want it to bind. And of course, use this for making all 100 different antibodies against spike, which was important as it relates to variants and how do you make kind of spike antibodies against the spike protein for invariant regions. So that's an important application. But we've made also antibodies against proteins that are not easy to make. So how would you design an antibody if you can't do the experiment? And computationally, you can do that. And so we've done quite a few antibodies now, or at least beginning to, that go after difficult targets to actually make experimentally. We've gone after certain aspects of signaling proteins. We have been working on, most recently, adjusting some of the gene editing machinery, again, using computational design. A very interesting thing is we've been training models to essentially embody or encode whatever it is about immune kind of reaction that is resident in the proteins you're trying to make so that you can make stealth proteins. Clearly, we don't make antibodies against self proteins, or at least we don't most normally. And the question is, could we train a system such that we could say, here's the protein I want to make, except I want to make it out of things that are not antigen presented properly. What can you tell us? Versus reasoning our way to this by incremental epitope maps, et cetera. So we're doing quite a bit of work on that. I'm not saying we've solved these things, but one of the things we've learned in working in this space and related machine learning spaces is that if you can get on the playing field, if you have a solution that is 10% good, the rest is further learning and further optimization. And so you shouldn't aim at solving the problem in the first instance. You should aim at solving the problem slightly and then realizing that more data, more iterative loops, more learning ends up with much, much better solutions. All right. And then the last thing we've worked on is completely coming up with a protein that looks nothing like the original protein, except does the same function. So kind of recoding it into a different set of amino acid sequences. So that's another thing that computationally can be done. All right. So this is just one of the interesting projects that has been developed. A second one that I'll tell you about is gene editing versus gene writing. So we started several years ago asking the question, really, is the only way of doing gene editing taking bacterial kind of immune defense mechanism of the CRISPR-Cas9 system and use that as the only way to edit mammalian cells? And of course, you know in human genetics that there's massive amount of mobile genetic elements that are in our genome. Potentially the highest number of genes that we have are estimated to be mobile genetic elements. And so there's a vast factory or reservoir of protein or gene editing tools that are in our genomes. We just haven't known how they work and exactly how to harness them and control them. So we set out to do that, also aided by some amount of machine learning, but also experimentation. So we asked the questions, could we take mobile genetic elements? Now, by the way, you may have since in the literature read about some academic work that's being done in this area. 5 years ago, when this project, now called TESRA, started, there was not a single proof of any retrotransposases or even recombinases that had been harnessed to actually make a human genome insert or major alteration as opposed to a single base alteration. We were interested not in single bases, we were interested in writing or rewriting large chunks of genes. And that's what this project set out to do. Again, I'm not getting into the scientific details given our talk and the time constraints, but just to kind of tell you that the general notion was to look at the repertoire of evolved parts and then from there try alternative approaches with the belief that such things could be done and then working backwards to see how you enable it to be done. We identified 75,000 potential DNA gene writers in the human genome. And by the way, beyond as well, we looked at other mammalian systems, et cetera, we thought could offer parts. As well, RNA-based gene writers, and from there, basically started doing these learning cycles to come up with potential solutions. And we've shown so far a set of capabilities in RNA and DNA writing and rewriting that we think makes it a lot further extended than just the editing, single base editing that we're hearing about. And we're on our way, we're by no means at the end of the road, on our way to, okay, what do you deliver? It's going to be an RNA-based, mRNA-based code. How do you deliver it? It's most likely initially LNP, could be in a packaged in a virus as well. And how are you going to manufacture it? So all the things you needed to figure out to make a protein code, you now have to figure out to make a gene writing apparatus, and we're actively pursuing that. We think if we do it this way, we'll be able to incorporate relatively large sets of DNA sequences very precisely into safe harbors within the genome, not the way a lentivirus or an adeno-associated virus would, and that's what's exciting us about this whole approach. I'm going to give you a third example, just to keep kind of flowing in different directions. One of the things that we learned through the Moderna experience and several others, like TESRA, Omega, another company in the epigenomic control space, is the difficulty in getting things into specific cells. And this is a problem that's being pursued in many different labs. What we started getting interested in is that we talk about programmable medicines in much of what we do because we're using code molecules. And the question is, does the body have its own way of programming delivery of things from one type of cell to another type of cell? And that interested us because we're prone to look for ideas or hints of solutions in nature as opposed to only relying on human ingenuity to come up with engineered solutions. And so while on the right side, we've enjoyed the kind of revolutionary potential of mRNA, which is itself an evolutionary technology, the question was, well, what about intracellular communication or intercellular communication? And there you realize that the body has invented its own nanoparticles. We call these many different things, extracellular vesicles, exosomes. They're all different things, and everybody has their own favorite name for different subsets of these based on size and function. But we said, well, why don't we just look at the massive compendium of these things that exist in nature, not just in humans now, but in plants, in bacteria, in fungal species. Just look at every extracellular vesicle, and instead of trying them all, because that would take a little time, to actually render them back into what lipids is nature using to do the same trick? And might it be that the use of the composition of those lipids offers tropism, offers other stability advantages over synthetic lipid nanoparticles. And that's what this project that's now called Senda Biosciences is pursuing. The notion is, can you make programmable nanoparticles coupled with programmable nucleic acid code in order to deliver more programmable at the body level medicines? And so we've begun to do that. Out of a set of some 55,000 different types of lipid sterols that have been identified that are used in nature to make extracellular vesicles, we've tried various combinations of these, quite different than the lipids that are used in synthetic particles today. And we've just numbered these on the top in the columns. And what we're just showing you for visual demonstration is that, in fact, these different things go to different parts of the body. You can have things that will go to the liver, and you can have things almost exclusively to the liver, and you can have things that barely at all touch the liver but go exclusively to the spleen. Or for that matter, you can look at things that are now going to the lungs that are, like if you look at this thing all the way to the right, goes to the spleen and lungs, but very little else. And so now you could imagine a whole set of these, but the components inside of these are actually all natural lipids. They're not synthetic compounds that the body has never seen before. So we're quite excited by the potential, it's a long-term journey, potential of doing tissue targeting and opening up a whole new set of capabilities. And then the last example I'll give, I want to make sure we have time for questions, is this project that's called A Priori. Here, what I just want to leave you with is the notion that, coming back to the vaccine and kind of pandemics, is the challenge now has gone from, can you make a single vaccine, or can you make one that goes after a couple of strains, to, can you make a vaccine that goes against every strain that are variants from an original one. And to do that, you can either chase the virus, or you can find vulnerabilities and make antibodies against those, or vaccines that evoke antibodies against those. Or you basically look at every single variant that's been seen in nature, and we've done this with COVID, and we've done this with influenza, or we're doing it. And from that, train an algorithm to literally come up with a net, a pathogen shield, a net, that would essentially capture not only the current variants, but any variant that are 5, 6, 7, even 10 mutations away from where you are today. And we were actively working on this when Omicron hit, and Omicron made a gigantic jump in the number of mutations that we saw, exceeding what we thought would have been something we have to prepare for, so it made us humble. Nature has a way of doing that. But the reality is that the computational approach can keep up with that. The experimental approach, the diversity you have to anticipate to come up with something that catches 35 changes, is an enormous diversity field. But it turns out that computationally, these are, we believe, tractable problems. So we're actively working on the ability to train through machine learning systems that will then suggest a minimal set of targets against which a vaccine may have effect, and we're going to test these out. A lot of this, by the way, is experimental in terms of making every one of those variants. These are not viruses we're making. These are just the spike protein or the RBD or whatever protein you want to go after, and then testing them in vitro first. This is laid out here, but in the interest of time, I'm going to skip it. Those of you who are interested, there's been a little bit written about this project, and Octavia, which is the machine learning system we've built to be able to enable this. I'm going to wrap up by just saying, looking forward, out of this last few years and out of the technologies that are coming out, look forward to immune medicines having a much, much bigger impact on human health than they did historically. If nothing else, the virus taught us how little we know about our immune system, despite knowing a lot about our immune system. And so the good news is, with 10,000 or 100,000, I don't know how many meta-archive papers have come up on the immune system precisely in every type of T cell and every type of antibody, that's going to have massive effect. And the tools that have been developed, computational, physical, to really make advances there. So we're very, very active and excited about this. Look forward to programmable medicines, not just programming based on RNA, but programming based on DNA, RNA, proteins, and everything in cells. And the advantage that I lay out here for what programmable bioplatforms really can do. I won't have the time to talk about that, but those of you who are interested in this, you can look at a technology platform in a project called Laron that is using now circular RNA and what we call endless RNA to make translatable proteins. Alterna, which is making tRNA-based drugs to essentially read through otherwise rare genetic disease stop codons that are misplaced, so that you can actually make a full protein. And experimentally in animals, we've shown the ability to do this with tRNA as the drug, synthetic tRNA. In the interest of time, I'm not going to take a lot of time, but just to tell you, yes, we think that a major new frontier is thinking about health and medicine in a preemptive sense, not as a reactionary treatment sense. If you take a step back, you'll realize that, I've been doing this type of work for 35 years, and I realize that most of my time has been developing heavy-duty weapons that all cost $1 billion to build, just like stealth U-2 bombers, this, that, and the other. And in fact, we do very little upstream because society doesn't reward it, governments don't allow it, regulators don't allow it, by and large. And yet, it stands to reason that if we could actually catch disease early, everybody knows that, but in fact catch disease before it's a disease, that is in a pre-disease state, and we can do something to delay it at a minimum or completely derail it and make it into something else, that we could potentially, for a lot less cost, have a much more profound impact. The problem is people tell you, prove it before I'll believe it, and you can't prove it if they don't let you do the experiment, or if the experiment takes 20 years. So there has to be something else that constitutes proof. Otherwise, we're going to be making really expensive late-stage disease weapons, and healthcare is going to go from what I was just told last week in the US last year was 19.5% of GDP, which is appalling. It's going to go to 30.5% by the time my kids are my age. It makes no sense. And I would argue that the same science, the same biomarker discovery, the same type of interventions we're doing late can be done early if there is the societal belief that we need to secure our health as opposed to resign ourselves to be getting sick and then hoping for magic treatments at the end. For those of you who are beginning of your career, reserve some space and time to innovate in that space. And by this, I don't mean supplements and things that are, historically not had the scientific backing, but rather the kind of things we do in biotechnology, but applied to very early disease. That, to me, is the enemy. If we can control the enemy when it's before it's become a real threat, I think we're going to go a lot further. And vaccines are exhibit A in that, and we just collectively saved a whole lot of lives based on the power of vaccines, and yet every other dollar in healthcare is spent not upstream of disease, but as far downstream of disease as is possible. And then the last thing I think just worth mentioning, and because it wouldn't be fair otherwise, is so many of the projects now are being enabled by, infiltrated by, and augmented by machine learning and artificial intelligence that as a biologist, you really don't want to be afraid of computation quite as much as I was and others today are, because it's really going to become integrated. It's going to become all an information science. And so how to think about the biological advances happening in concert with the ability of algorithms to represent the complexity, or at least embody the complexity, even if it's not human understandable, I think that's going to be a major additional frontier. And this, just to leave it off in a fantasy where the whole thing started, this is kind of the modern day fantasy that we're playing around with, which is, could we actually train machine learning algorithms, and we're actively working on this otherwise for years, otherwise I wouldn't tell you, to actually identify the kind of leaps that otherwise we now make through the teams we've built and the training we have. And that is, could you imagine futures using AI trained systems that are not connectable to the current present scientifically, but that could lead to this reverse type of discovery that I talked about. And so the question is, can you create projected futures and use that to do the kind of innovation that we would like to do? And we call this kind of an AI biologist approach. And we think that the kind of language processing, data processing, generative capabilities are going to enable that to happen. Initially, it's going to be really, really crude. It's going to get every 1 out of 10 projections are going to be actually understandable. But we think once we are there, after that, it's just turning the crank, as they say. And I think that's going to be a very interesting adventure to follow. I think that's about it. So with whatever time we have left, I'd love to take some questions. Thank you so much.
Thank you, Noubar. That was a tour de force, astonishing presentation, and very, very different than it was 6-7 years ago. Every slide had AI underpinned. I mean, 2 questions I'll start with. One is, if any of us in this university submit a proposal to one of our research councils with this type of crazy thinking, I don't think we will ever get funded. So how do you, it's not just the thinking out of the box, but how are you managing to fund such high-risk, high-reward ventures in the US? And I suspect you have sort of close working relationship with MIT and Harvard and other organizations nearby you.
So yeah, it's a great question, and the answer I'll give you is a bit unfair and not representative, simply because after trying to do that with academic collaborations for many years, about a decade ago we stopped because we could not, in fact, we could not figure out how they're going to get funded to do this kind of work that we thought was important. And so we just set up our own ability to do it in-house.
Yeah.
So today, out of the 300 people within Flagship's core innovation team, about a third of them are MDs, PhDs that practice this kind of innovation entirely from capital that we have sourced that entirely goes to our own internal inventions and internal innovations. And so we recognize that if the first thing you have to do is to convince somebody to give you money around your ill-formed idea, far-fetched idea, there's nowhere to go.
Okay.
And so you need the money before you have the idea.
Yeah.
And so how do you get the money? And that is you basically point out to people in the first instance that most great breakthrough innovations did not come from really well-formed ideas that were proximal to the ultimate thing, but came from this other place first. Second, that they shouldn't take every dollar they have and put it into this, but maybe $1 out of $100, $2 out of $100 ought to be used to enable this kind of innovation versus not. Over time, we've amassed quite a bit of experience and history in being able to do this type of work, and so we were able to attract that kind of capital. Just to give you a sense, this level of activity that I described a few samples of, on an annual basis, we launch about 6 to 8 bioplatforms that had no precedent in any literature before a year.
Wow.
And the 6 to 8 combined occupy $300 to $400 million of spending a year to give rise to them collectively. And the broad community of companies that have come out of this work that exists today in Cambridge is about 6,000 people. They spend collectively probably almost $5 billion a year now in research. Across them today, there's about 55 drugs under development and several agricultural, sustainability products, et cetera. So if you wait long enough, you get this to a scale where it kind of feeds itself. But academic work in this regard, I mean, frankly, the biggest interaction we have with our academic colleagues is that they come along on these journeys with us as private citizens, meaning they love this kind of ability, but in fact, it makes it easier because if they do this in their own labs, there'll be a bit of a conflict of interest. But if they just do it on this other journey, they are fantastic thinkers and rigorous with what questions we need to ask, what things you have to believe. So we have a lot of academic ties from all over the world, but it's not taking science that's just been reduced and then converting them. That's not the kind of innovation we're doing.
Fantastic. Great. So, Hutan first, yeah.
Thanks, Noubar. Very eloquent epistemology of emergent discovery. And in terms of practice, evolution has a behavioral element, the Mark Baldwin effect. And that phenotype first approach in terms of having behaviors of learned innovation is something that can be maybe augmented. How do you see that panning out, both in the commercial sector but also in the academic sector?
Well, for all of you who know Baldwin effect extremely well, which might be 3 of you, I know Hutan does. Baldwin effect is this notion that kind of was around the same time as Darwin. I think it was a contemporary, or maybe soon thereafter, where there was evidence shown of something that would otherwise have been thought of as Lamarckian and therefore definitely frowned upon, but was actually evidenced in nature. And that is that genetic changes coupled with learned behavior could give you a gain of function that over time, through descendancy, would find its way into genetic changes only because you couldn't count on the learned behavior being passed generation to generation. And so you didn't need to solve a problem purely by getting a genetic solution to it, that you could find the problem by having a phenotypic or otherwise maybe even epigenetic variation long enough that it makes its way all the way. That was the basic general idea. And so in this kind of evolutionary kind of approach, that happens a lot. That's what the learnings in our teams are all about. Because before they will find a technological solution to something, they have to keep it alive even though it's not solving for something that's valuable, long enough that the solution underlying it, the platform, will actually find a way to enable it without the belief system. So the notion of faith, which is belief without facts, well, that is a form of Baldwin in the form that it's behavioral, and that behavior enables things that would have otherwise been long dead to persist long enough that actually the solution comes about, in which case you don't need the faith. And then people in newspapers say, oh, I had invented this 20 years ago, or everybody can do this, etc. But the reality is that bridging of the partial genetic solution to an ultimate, but you know, I mean genetic, I mean like kind of, so it is a big part of kind of a community of practice that, so of course, the other thing you have to do behaviorally is to not judge defective ideas as being predecessors of terrible ideas, because if you do that, you'll get rid of them. That's another belief system. So it's belief as to what's possible, but also belief as to what to preserve. Otherwise, you're going to come up with fairly limited solutions. There's a bunch of things that we've learned over time. We've tried to, there are various things online that kind of describe these approaches. For those of you who are interested, there's different talks we've given and stuff that goes into the mindsets it takes to do this kind of work.
Thank you. Yes, sir. To introduce yourself. Yeah.
Ahmed Nabil, I'm a PhD student at IGHI. Thank you for the inspiring and very informative talk, and congratulations on your extraordinary journey. And thanks to Prof. Darzi for organizing this event. I have a question of two parts. Have you ever faced throughout your journey a dead end with some of these what-if questions, and how do you justify it internally? The second part of the question is, if the answer to these what-if questions is yes, we can, how do you deal with the resistance and adoption?
Well, so first, we've run into many more dead ends than we've run into live ends. And if we didn't run into dead ends, we would be convinced that we're not trying hard enough. That's the kind of the definition. The feeling of running into dead ends is either that you're working in a really crowded place. That's what the adjacency oriented innovation often runs into, because somebody else got there before you. Or it could be a dead end because it's just not either potentially valuable or physically kind of realizable. So that we get a lot of that. And in fact, one of the things we learned early on is we needed to create a compensation mindset that rewards, that gives people their own ability to distinguish the dead end from a live end, because their own progress and their own reward system depends on that. So one of the things I would argue is that in a traditional startup environment, when there's one idea, death is a pretty harsh end result. In a situation like this where many of our teams, all of our teams are working on multiple different things, then your opportunity cost is pretty large to persist in something that otherwise may end up in there, so that when you're seeing objective evidence, rather than what we call in the States drinking your own Kool-Aid and believing, there's a limit. But one good way to do that is to have people say, you know what, I'm going to spend some more of my time enabling this. I'll keep this maybe at a slower rate until some data emerges. So we let a lot of the scientists and the project leaders kind of make their own trade-offs. So that's kind of what we've learned. How do you create adoption? That's a tricky thing, especially when you don't have a lot of data. And so one of the things is you want to be very straightforward and honest when you're actually telling people something that isn't real yet. And it turns out that people can use their own imagination to imagine a version of what you're saying that might be valuable. But if you try to sell it to them as though it's real, when it's not, they'll lose faith and confidence in what you're saying. So there's a level of, you can be honest about what is real and what is not real, as opposed to thinking, I got to make everything look real, otherwise they won't believe me, because ultimately they'll know, and then you'll lose your reputation. So I think that the average person will not be willing to make that same leap of faith you are. But you're not looking for everybody to believe. You're just looking for a few people to believe. And the few people who believe tend to know that they're not going to get outsized returns unless they take outsized risk. And one outside risk is to get involved in something before it's known to be real. And those are the people you're trying to detect. Those are the early adopters.
Thank you. Thank you. George, Professor George Hanna.
Very inspiring talk. I'm just interested to know your thoughts about the processes for killing projects early. Like you do quite a few projects, some of them will be wonderful, and some of them you will stop. So when do you stop them, and what's the processes? just some of your thoughts about
Yeah. So because the same people who are coming up with the what ifs and doing the actual work are the people who stop them, versus having a central committee who reviews them with gray hair, etc., and shuts them down. We have no such function. So we do not shut down projects that the team thinks is actually on a good path. Now, to do that, you need to have a large amount of money to be able to keep things proceeding. So we don't have a filter that is different than the actual process that I described. The evolutionary process leads the teams themselves to conclude that you know, if I said, what do you have to believe this can be done? And if my answer is 1 minute long, that's one thing. But over time, if the answer becomes 5 minutes, because I have to say, this has to happen, then that, then if this happens, that, the longer your explanation, the more you can even tell yourself, listen, it's 2 minutes, the compounded yield, you learn as a chemist long ago, or early in your life, that 10 reaction steps with 95% yield gives you a few percentage at the end. Well, it's the same with these things. If you have 10 things you have to believe, each one you have a 95% certainty in, the compounding is a pretty big problem. So we try to get the teams themselves in an evolutionary selection way, not in a screening, filtering way, to actually come to these conclusions themselves. But the other thing that I didn't mention that I just want to kind of get people to think about is we describe projects in a reductionist way, as though it's one idea, one approach, one company, this. But in the way we're thinking about these things, any given what-if question may give rise to 5, 10, 20 different, kind of like a cloud of solutions that you initially can envision, which if you've compressed them will come from, let's say, 3, 4 fundamental alternatives that then lead to many different alternatives, right? So the question becomes, how long can you keep the cloud alive? Because to use a crude analogy, just like in tumors, where you have heterogeneity such that when drugs hit, the parts that are resistant gain advantage and they proliferate, and it looks like the cancer gained resistance. It did no such thing. That variation was what allowed it to do that, or viruses. Similarly, in this, if you can keep multiple instances, an mRNA that is actually has to go into a tumor and that is doing this versus a vaccine that is going to draining lymph nodes versus, you know, there are many different possibilities, all stemming from the same underlying idea. My advice is keep multiple of these alive, because what will be killed are particular branches early on, not the whole thing. But if you only have one instance of it, because you were very clever and you reduced it down to one thing, chances of dying are infinitely higher, in my experience. So one defense mechanism is think loud, think like an evolving thing, and make sure you have heterogeneity or diversity as long as you can in the journey. Now, that adds capital to your needs, but it ensures survival, I've seen empirically, much longer than if you reduce it to one and you have just enough money for that and then you're dead.
Great. There is about 100 people online. Do we know, do they have any questions, or could we figure that out? Or they're not really, they can't ask questions? Okay, great. So
They can just wonder.
just wonder, yeah. Okay. I'll go in the middle
I'll
there
give shorter
and then come back. Yeah, please. Introduce yourself first. Yeah, thank you.
So you were talking about having this wonderful research team at Flagship who are coming up with ideas and iterating on those things. And I imagine that's very expensive to run. If you are a university and you're more interested in promoting this sort of evolutionary thinking, how can you interact with your funding sources to convince them to support these sorts of projects, to say, actually, you're not going to see that many successes, you're going to see loads of failures, but we still want the money nonetheless?
Thank you. Let's take another question, because there's a similar question we've asked before. The gentleman there, yeah.
Hi, thanks firstly very much. That's a really interesting talk. My name's Oscar Marshall. I'm a PhD student in ChemEng. I'm just thinking about that slide you showed from the leap of faith to the growth company, and I was just wondering if you could expand on the time frames that each of those individual processes tend to take. I'm sure it's different between different projects, just around about.
Mm-hmm. So in a nutshell, the explorations could be a couple of months to sometimes years, because they kind of are in the background. If we're actively doing an exploration that's leading to a protocol, that could be a couple of months of expert iterations, etc. So in that range. But once something becomes a proto company, that's usually actually highly prescribed. It's about $2 million and 9 to 12 months. And what that does is it limits the range of projects we could take on to things that for $2 million and 9 to 12 months can be reduced to practice. So we don't work on, you know, laser fusion. We don't work on things that are going to take 20 years to show, because we'll wait for 2 years before they can be realized, before we will make the leap of faith, if you think of it that way. So for example, in 2002, we started a company that was earlier than what the words synthetic biology were used, a company called Codon that actually made, or Ingenios actually stood for Engineered Genomic Operating Systems, that ended up being a predecessor to synthetic biology. We found out that we can't actually print DNA long enough pieces to put into cells at the time, and we had no idea how long it'll take to develop that capability. So we just paused it, waited 3 years, and started another company called Codon that did the same thing, except now we were proximal to having that. But our advantage to other people was that we knew exactly what to do, but for that technology existing. So it's an interesting thing where you can actually be poised and frozen. But so to answer your question briefly, a few months in exploration, if everything goes sequentially, 9 to 12 months in a protocol, sometimes it runs a little over time. And then the nuclear phase is 2 to 3 years. And it's all dedicated to platform building. So we will not rush into with an idea, the first protein that we make with Generate, we don't put into the clinic. We actually give ourselves 2 to 3 years of platform building time. And then in the growth phase, so it's about 3.5 years after you start this journey, maybe 4, you're now in a growth where you're actually trying to come up with product pipelines. So it's a fairly lengthy journey.
Great. I think I'm just going to give the last word to our king of RNA, and that's Robin. And any thoughts about what you heard? And, you know, Robin has been, just to say, without embarrassing him, he made probably the most significant contributions to mRNA vaccination during the COVID pandemic, and we're very proud to have him here. Robin.
I'm not sure that that's entirely correct, but I mean, I thought your talk was very inspiring, but one critical component is about resources. And so evolution has that pressure of fighting for resources. And what you've built up is an engine where resources aren't limiting the number of ideas. And I think that that's critical to success. And actually looking at our own experience in the mRNA space, where as an academic university, we went through the same journey in terms of design into development within a matter of weeks. Where we ran out of steam was the lack of a pharma backer that could give us the redundancy to look at a wider range of, for example, formulations that would have actually meant we had success. So how do you ensure that you can keep enough funding outside of what you've built, if you were starting today, to keep ideas alive?
I mean, yes, this was a similar question that's come up, and I think it's if I had a different idea, I'd be trying to convince other people to do this. There's such a kind of dogmatic belief in the way we fund innovation today that it's basically experts decide what's the next adjacent reasonable thing to get funded, and then that's the only thing that gets funded. But I always remind people that the people who decide what gets funded today didn't used to get what they wanted funded early in their career. Then they end up in the front of the line, and they kind of perpetuate that which they really found problematic and limited their career. Bob Langer, who's a famous MIT professor who's prolific inventor, will tell you that everything he applied for got turned down with angiogenesis, with drug delivery, whatever. So I think that I'm not skirting the issue. I don't know how to make systemic change. I have to believe. And by the way, the reason I talk about this, like what benefit do I have in talking about this if it's going to frustrate people who can't get access to capital to do the same thing? Answer is, at some point, people are going to say, why don't we take some of our central resources and dedicate 5-10% of it to that? Why doesn't the NIH do that? Why does the MRC do that? And you know, Wellcome Trust can do that. And by the way, when I talk to them, they tell you, we do that. We do a lot of that. No. We have all this. But the reality is that they do it reluctantly, subject to experts telling them it's really a good idea, those are not the experts that should tell you what's a really good idea to make a leap in. So I think what I can do is to give examples, to create caution that this is a messy process, breakthrough innovation in general, that we should not believe the stories that get told after breakthrough innovations are made as the way in which they get made, because that misdirects people to think they have to find geniuses before they become geniuses. And I can tell you, 35 years into this journey, I completely distrust that. You can call people whatever name you want to give them. They're not geniuses in the rough when they're making these. They're part of a participatory, collaborative process that maybe they had the propensity to jump a little longer than somebody else, but at the end of the day, it's a group activity. And I think once you realize that, you want to create that. To your point, though, you made a very important point I think the audience should understand, because I often get asked, what if we poured a lot more money into startups? Would we get better startups? And my general answer is no. Because in fact, if you believe in the evolutionary way this happens, you'd realize that resource constraints are what drive competition that then causes the kind of surviving environment in which advantage is gained from an evolutionary stance. So if you poured a lot of money, every species in that cluster can survive, and there's no reason to compete. You're not going to get a gain of function. However, to the earlier question that I said about creating a diversity of your ideas all alive, turns out that if you have one idea, right, then you need a certain amount of money. If you want to keep 20 versions of the idea alive, even the large amount of money is not enough. And I like that because you're still feeling the competitive constraint because you feel like you don't have enough money. Does that make sense? Yeah. Except we've traded the extra money is going to preserve the resilience of the overall idea against lots of unknowns versus a fundamental bet of overfunding a singular idea. And yet, when you go to large funding sources, they like to find the singular idea that they want to pour a lot of money on because they can gain consensus. People say, that one, 100 people apply, 5 people are finalists, we chose that one, we pour all the resource into that one, and it could be a huge blow up or not. I'd rather fund a bunch of related things, keep them coexisting and co-developing, still feeling constrained because even a large amount of money doesn't fill those. That's evolutionarily consistent, I think.
Great. Okay, so could I please ask you to show your appreciation to Noubar's time and coming over to Imperial and